Hey, welcome back for chapter 25. This is the last chapter in exam 2, and this one's actually pretty straightforward, so let's get on with it. So this is DNA structure and gene expression, and about the mid 20th century, we discovered that DNA is the genetic material of life. Contrary to what everyone seems to think these days, Watson and Crick did not discover DNA. We knew about it long before them. What they discovered was the three-dimensional shape of it, and they actually kind of stole half of that research. So, yeah. But anyway, in order to be genetic material, you have to be able to store information. It has to be stable, and you have to be able to replicate it with high accuracy. So, James Watson and Francis Crick determined the structure of DNA in 1953. They did not discover it, as previously stated. DNA is a polymer of nucleotides, and nucleotides are the monomer of DNA. Makes sense, right? So each nucleotide is composed of three parts. You have your sugar in DNA, that is deoxyribose. In RNA, that is ribose. You have your phosphate and your four nitrogen-containing bases. Two are purines, two are pyrimidines. Your purines are adenine and guanine, and your pyrimidines are thymine and cytosine. Now here's the only tricky bit. In RNA, you replace the thymine with a U. So the way I keep this straight is that pyramids cut, because C for cytosine, U for uracil, T for thymine, and it's your pyrimidines, which kind of sounds like pyramid. So, or you can think of it, Ag is the atomic symbol for gold, so pure gold, purines. Either way you keep it straight, it's fine as long as you keep that straight in your head. And then you have to understand that A bonds to T and G bonds to C. So you can kind of think of it as the two curvy letters bond together and the two straight ones bond together. The only catch is, again, you have to remember in RNA, you swap out the T for a U. And these are hydrogen bonds that hold them together. You've got your phosphate backbone here, your sugar phosphate backbone, and then the rungs of the ladder are your nitrogenous bases held together with hydrogen bonding. When you replicate DNA, you have to unzip it here, and then you unzip the two strands of your helix so that you can get in here to the middle of it to access it, the base pairs to replicate them. And this is a semi-conservative process, meaning that the two resulting daughter strands are each half new DNA and half old DNA. There is some evidence to suggest that in certain stem cells it is a fully conservative process, but that has not been proven yet. It is merely anecdotal. So for our purposes, semi-conservative, meaning each daughter, which is identical to its parent, but it has half of the material from the parent, and half was newly synthesized. To replicate DNA, you have to unzip these two strands using DNA helicase to open it up, and then you take new complementary nucleotide sequences and position them with DNA polymerase, and then DNA ligase breaks any or repairs any breaks that happen in this process, because it's kind of like unwinding a slinky. You're going to have a lot of stress put on the backbone, and it does frequently break during that process, so you use ligase to put it back together. And then once you've finished with that, then the two helixes are identical to their original parent, and it's just kind of as you're going through, helicase is creating a area that's unzipped, and then as you seal it up behind it, or as it moves through, then it wants to pair again behind it, and you end up with this one being unspooled and two daughters coming off of it. Which is all this is saying right here, basically. RNA is a little different because you use uracil instead of thiamine, and you use ribonucleic acids instead of deoxyribonucleic acids. Because the sugar is ribose instead of deoxyribose. You have three main categories of RNA. You have your messenger RNA, which is produced in the nucleus during transcription and takes a message from DNA to the ribosomes. Then you have your transfer RNA, which brings amino acids to the ribosome to build the proteins with. And then you have ribosomal RNA, which is produced in the nucleus and makes up the ribosomes along with some proteins. This little chart right here basically summarizes all the differences and commonalities between DNA and RNA. I can pretty much guarantee you there's a couple of questions drawn from this. So gene expression, we have a couple of ways of regulating this. You can either regulate it at the transcriptional level, meaning do you make a copy of that in the first place? Are you transcribing that DNA into messenger RNA and having it do its thing? Or you can regulate it at the translational level, which takes place in the cytoplasm, and that's basically how many copies of that gene or of that protein do you make per copy of RNA that was sent from the nucleus. So during transcription, you have segments of the DNA that serve as a template for the production of RNA for all three classes of RNA. Then you have RNA polymerase binds to a promoter sequence in the DNA. The helicase opens up the DNA so you can do your base pairing, which so far this is fairly similar to replicating your DNA. The difference is you're producing an RNA copy of a specific section instead of the whole thing, and you're using RNA polymerase instead of your DNA polymerase. So far so good. Once you have copied your RNA here, and you've transcribed your RNA, then you have this processing of your messenger RNA, and you have, and then kind of need to zoom in here a bit, hopefully without breaking anything. Okay, so you have your DNA template, and you make your primary RNA sequence. This is not the final thing that gets sent to the ribosome, because you have all of these introns stuck in here. And you cut these out, and then you assemble your final RNA. So it is actually the exons that are expressed. And this is part of the reason why you can have so much variability coming out of a single sequence of DNA, because you can, at each of these exon or introns here, when they're cut out, you can rearrange the order of things in some cases, and you can get a lot of proteins out of one single sequence of DNA. So this is just practice here for you, practicing transcribing DNA to RNA. Just remember that you don't have thiamine in RNA, you use uracil in its place. Again, more practice here for you. I can pretty much guarantee on the test you're going to have to do this at least a couple of times. And you need to understand that you transcribe, then translate. And if you're going to go from here, if it asks you what your transfer RNA sequence is from this point, you have to do your base pairing again, essentially. Transcription expression, or I'm sorry, gene expression. So we've done our transcription, now we're going to translate. So this is where you get your protein synthesis going on. You've got your triple code, the triplet code, which is 3 nucleotides of messenger RNA, forms a codon, which you can make 64 different combinations of that, but we have 61 amino acids, which means there's a little bit of redundant wiggle room in this. So a single point mutation, odds are you're just going to end up like mutating it to there's a chart you'll see later on. There's like 4 different things that code for cytosine. So if you mutate that one particular sequence, you're still just getting cytosine. So you have some protection against mutations there. You have 3 stop codons, UAA, UGA, and UAG. Definitely know these. And also later we'll see the start codon. Definitely know that one. But you can see in this table here how you get your code. This table is for the code. The AUG is your start right here. That's a methionine, but that is the single most important one to know, as well as your 3 stop codons. And if memory serves, there's like 1 or 2 questions. I think it's just 1 in the Q bank that actually expects you to be able to take a DNA sequence, take it through to RNA, and then figure out what protein that is. But it's like really common ones like proline and theanine. So don't worry too much about trying to memorize this one because it's just the 1 question, but it is there. So this is your overview. This right here kind of puts everything into perspective for you. You've got your DNA helix right here. You pop that sucker open and you transcribe that, which takes place in the nucleus, to your messenger RNA. The messenger RNA, you can see here you've got your reading frame with your 3 base pairs per codon, and then that gets transcribed at the ribosome. And here's the thing, you have your anticodon in the ribosome for your transfer RNA, which bonds to a particular amino acid and puts those in order. And it's an anticodon because it is bonding, to the codon. So it has to be the opposite sequence of it. A to G, T to, A to T, G to C, so on and so forth. And then you have your polypeptide, your protein down here, with your amino acid base pairs put together. So again, this right here is practice for you. You take your DNA to RNA, and then you make your amino acid sequence from that. And you do not start until your start codon, your AUG, and then you stop at the first stop codon that you reach, so UAA right here. So if you can do that, go through and practice that a few times, you'll be in pretty good shape for this portion of the exam. So we're going back to gene expression a little bit here. You've got your transfer RNA, which transports amino acids to the ribosomes, which then they bind at one end on the anticodon right here with your amino acid, and you put those in order based on the triplet pair, and that will produce your polypeptide, which I think there's a better picture. Yeah, right here. So you've got your small ribosomal subunit, large ribosomal subunit. They sandwich around the messenger RNA right here, and you in your A site, this is where your transfer RNA, which is a little green thingy here, with its amino acid stuck to the top of it, binds initially. If it has the correct anticodon to match to the messenger RNA's triplet right here, then it will proceed to the P site where it attaches its amino acids to the previous one in the sequence, and then at the E site it is ejected. If it is not the correct codon right here, then it just bounces out, and you just kind of repeat the process randomly until you find the right one in the next sequence, which is, you do that over and over again until you hit your stop codon, and the stop codon binds to your release factor right here, which does not have another amino acid on it. So as this kind of like works its way down, and then this final one here gets disattached from the chain, booted out, and ejected, and you have now released the completed protein and your your messenger RNA. You have on the ends of your messenger RNA here you see these little sequences where these poly A tails, and the more A's you have on the end, the longer this will last, because the poly A protects it from being digested by lysosomes. And if you run out of A's, then this is degraded. So that's one of the post-transcriptional ways that you can regulate the expression, because this thing can just go and reattach and start making another copy. But when you run out of A's, then it gets broken down and no longer is expressed. Which all of that is summarized in this nice diagram here for you. Your DNA gets unzipped, you make your messenger RNA, that gets sent out to, which is your transcription, that gets sent out here and is translated at the ribosome to proteins, and you're using your transfer RNA to bring your amino acids in to synthesize your polypeptide, which is your protein, and that's the process in a nutshell right there. Housekeeping genes, basically, they're the things that govern how active something is. So only a particular cell type will use the housekeeping genes in a particular function. So, for instance, your muscles don't sit around here using the genes for hemoglobin and insulin, right? But they all have the housekeeping genes, because that's what's telling them how to regulate which of these things are active. So basically the housekeeping gene says, you are this type of cell, these are the things you do. A gene mutation is permanent change in the sequence of the bases of the DNA, and they can either do nothing at all or completely inactivate the protein. Some cause cancer. Like, some mutations will lead to cancer. Some have hereditary effects and can be passed down from generation to generation. But very rarely you will get a useful and beneficial mutation. There's a particular sequence of... let me just open this real quick. It's easier to illustrate this way. This is a CD4 cell in your immune system, and it has this CD4 receptor right here, and there are some people who have a mutation to this. It's roughly like 10% of the European population, where the HIV virus cannot bind to it. And it's thought that originally this mutation was selected for because it also provides resistance to smallpox. But if you happen to have been born with 2 copies of this mutation, then you are effectively immune to the AIDS virus. So every so often you get something useful like that. Most of the time it's highly detrimental and causes cancer. These can happen through errors in proofreading. Basically, the DNA polymerase goofs. Or I'm sorry, when you are laying down your sequence, there's a goof in it, but the DNA polymerase proofreads and checks its own work, so that usually takes care of the errors in replication, but it can happen. Once every few million or billion base pairs, it slips up. You can have exposure to mutagens, radiation, or organic chemicals that cause damage to DNA. Chemotherapy drugs do this. They shred DNA. And the reason that kills cancer is because it's rapidly dividing cells, so they're more susceptible to DNA damage. Basically, the goal of chemo is to kill the cancer before the chemotherapy kills you. And we also have transposons, which are referred to as jumping genes, little bits of DNA that move from place to place and produce novel protein products. We use these mostly in our immune system because we have to develop specific antibodies against antigens that we've never been exposed to before, and by rearranging the sequence for it randomly, eventually you find something that works well enough to bind to it that does not also bind to your cells, so then you can have an antibody against that thing. And then you have your plasma cells that just switch over, or your beta cells that become activated plasma cells and just sit there and produce that antibody for decades. Like, that's what they do. And they produce that antibody. It's the memory cells that hold on to that, so they can activate other B cells to turn them into plasma cells later when you need that antibody again. You can have base substitutions. So you accidentally drop in, the wrong base pair here. This is a missense mutation, and it will change the amino acid around. You can have, so instead of making like a lysine, you make a phenylalanine or something. You have nonsense mutations where you do the same thing here, but what it results in is you put a stop codon here by mistake. Like, you change this base pair into something, and so it makes a stop codon, so you end up with a truncated gene product. You can have a frame shift mutation, and this is either because you've accidentally inserted an extra base pair here, or you have deleted one, but it still stays in its 3-letter reading frame. So the cat ate the rat. Well, you accidentally delete your C here, and then suddenly the rest of this doesn't make any sense. I mean, yeah, you'll randomly get something down to that does, but this is a completely different sequence now because you've shifted your reading frame over by 1. And you can either do this by 1, 2, or 3. If you shift it by 3, you've basically just inserted an extra amino acid and everything else is fine. 1 and 2, obviously, shifted by, 1 or 2 places. And that is it for chapter 25. So if you have any questions, comments, concerns, exam 2 is coming up really quickly. So let me know sooner rather than later, and we're going to go over all this in class. So be ready to discuss, and I will see you next time.